Mr. Comet, and Dr. LaGrassa. We're going to look into the effect of prolonged valine deprivation on mTOR activation. So I'm Kevin, and my mentor is John Blennis at John Blennis Lab, Pharmacology at Weill Cornell Medicine. So mTOR is mammalian target of rapamycin. It's a major regulator of growth in animals and controls most anabolic and catabolic processes in response to nutrients and nutrients-induced signals. The second keyword is valine, which is one of the essential branched chain amino acids and must be taken in by food so your body can produce it. And the third key word is PI3K/AKT, which is a pathway involved in cell growth and proliferation and is activated by growth factors like insulin. And the final keyword is RAG-GTPase, which is a regulator on the lysosome involved in cell growth and is activated by amino acids. So the big picture, mTOR is a regulatory protein and is divided into mTOR complex 1 and mTOR complex 2. So, alright, this one is mTOR complex 1 and mTOR complex 2, and they regulate cellular processes like gene expression, protein synthesis, and cell division. mTOR can be activated through growth factors like insulin, as shown with the yellow dot, through PI3K, and which activates mTOR complex 1, and amino acids, as shown in the gray dot, can also activate mTOR complex 1 through RAG-GTPase, and also PI3K also activates mTOR complex 2, which in response further activates PI3K. And since overactivation of mTOR is seen in over 70% of cancers, so like understanding the regulatory mechanisms in mTOR may be vital in the treatment of cancer. And so since many amino acids play a role in mTOR regulation, I want to see the effects of prolonged valine deprivation on mTOR activation. I hypothesize that prolonged valine deprivation will cause an initial decrease in mTOR activation before the reactivation of mTOR. So methods. First step, we'll have mouse embryonic fibroblasts will be introduced in a solution of DMEM supplemented with fetal bovine serum and other nutrients. And there'll be two samples. One will be allowed to develop under ideal conditions with amino acids, and the other one will be without valine. And then after at certain time intervals, certain cell samples will be collected, and metabolite profiling will be done in order to determine the level of mTOR activation. So this is if everything goes as expected, this should be the expected result. So with valine, it's constant mTOR activation over time. However, without valine, we should see an initial decrease in mTOR activation since amino acids aren't present to activate RAG-GTPase. However, the absence of amino acids should activate PI3K, which will cause a spike or reactivation of mTOR before there's a decrease. So it might be significant in treating cancer since many novel cancer treatments involve depleting cancer cells of essential amino acids like valine. So the fact that mTOR is reactivated under valine deprivation may add another level of complexity that scientists need to consider before employing drugs that restrict amino acid absorption. And that's my presentation.